Hello, everyone. It's really wonderful to be able to present. And as I'm getting going in the inherited retinal diseases section, I thought I'll start with a definition. In my mind, inherited retinopathies are a group of disorders caused by single gene defects acting in or on photoreceptors or in or on RP cells. There is obviously a homogeneous expression often across the retina of the mutant protein or the enzyme, but we all know in the retinal field that some retinal regions degenerate earlier than others. And there is certain areas that are relatively healthy and certain areas that are relatively severe or very severe at any given time point. And both cell autonomous and cell non-autonomous and stochastic and developmental effects probably all combine and overlap to produce these spatial distributions of disease that we see. And we are in this talk, as well as in general, very interested in the transition between the healthy and the disease. And we'll go in further into it as to why that is. In these en face images, whether it's autofluorescence or fundus photos, you can easily see often the healthy areas from severe disease. However, we are very interested in photoreceptors, and OCT is obviously the one modality that allows subcellular evaluation and quantification of subcellular features. And among all the subcellular features, the one that is of greatest interest to us in general is the outer segment where vision starts. And outer segment is on the OCT, as you know, between the inner segment, outer segment junction, which is highlighted with red here, and which is also by others called ellipsoid zone or EZ. And between the ISOS line, the red line, and the RPE would be the location of the outer segments. When we can see in this example of an RP patient from the fovea going out to about 30 degrees in periphery, there is a central region of well-laminated retina that is very healthy. There's lots of outer segments, and it quickly changes to an area of great disease with very few, if any, photoreceptors and certainly no outer segments. We are calling these two regions definitely present outer segment versus definitely absent outer segment, and borrowing the concept from autofluorescence, which is uses similar concepts. And here's another example, which is less known, I believe, in the field, in another RP patient. And again, there is a central region of healthy retina and peripheral region of more diseased retina, certainly not as diseased as the one that I showed before. And in between is this ambiguous transition zone where the inner segment, outer segment line with the red becomes more and more difficult to see, to be visible, and it disappears. And we call that region QAOS, or questionably absent outer segment, where things are ambiguous. And we are very interested in this ambiguity, region of ambiguity, and what happens to that over time. So what we set out to understand is what is the spatial distribution and the temporal progression of these retinal zones of ambiguity that we are calling questionably absent outer segments, QAOS. To do that, obviously, we need spatial information from the OCT, and this is commonly done with raster scans or volume scans or 3D scans. Everybody seems to have a different name for it. And they usually cover the macular region, 20 by 20, 30 by 30, and that is fine, except that this is what the disease looks like in our hands. This is a set of patients at different severities, from least severe to more severe in this group. And certainly in cases of greater severity, where the retained region is delimited to the center of the macula, the macular scans that I showed is very well explains nearly everything that we want to know. However, in milder diseases, we need to go further to understand better, because the boundaries can extend beyond the macula, or there could be this moat of cell loss separating the macula from the periphery with a peripheral retention. So for that, we use ultra-wide OCTs performed by digital mosaicing. The way we do that is we get a whole bunch of raster scans extended across the macula with the aim of registering them and forming them into a resampled map, hoping to get about 55 degrees across and 55 degrees in the vertical direction. And they can be resampled isotropically at 0.2 degree resolution. And these pseudo B scans can be performed in either horizontal, vertical, or diagonal directions to be able to look at the spatial distribution of these unusual forms of transition zones that extend in different directions and have different forms. The next thing we need is we have a lot of data, and it's very time-consuming and sometimes impossible to do that manually. So we formed a two-level artificial intelligence framework. The level one is a typical CNN network that looks at each LRP. So these are the reflectivity profiles across an OCT looking into depth. And at each location, it outputs the probability between 0 and 1 of whether the ISOS peak, this junction between inner segments and outer segments, is present or not. So this probability input in a spatial region around the center point is fed into a second level artificial intelligence network, which is based on a multi-level perceptron, MLP. And it inputs these probabilities, looks at the spatial region around it, and outputs a quaternary map or a quaternary result at each pixel. And the three of them is the three outer segment disease criteria: definitely present, questionably absent, or definitely absent. Or if it cannot decide between them, it provides an output that is unclassifiable. So for an artificial intelligence, of course, one needs a training set. Here's an example of a training set where there is a central region of retention, peripheral abnormalities. And if one looks at a single location in its immediate neighborhood, one can see that there are regions of definitely present outer segment that is defined, annotated by an expert. And outwards, there's areas of definitely absent, and in between some regions which we are calling questionably absent. So once we feed all of this in and feed all of the data for patients, here's an example of a result we get. On the left is autofluorescence image showing these boundaries where the RPE is transitioning from health to disease. And in the middle is the output of the AI with the OCT showing where the outer segments are healthy, diseased, or ambiguous with the gray, black, and the yellow, and a single scan vertically showing what the OCT may look like. And what we may be able to observe is that some regions of these transitions correspond quite well to autofluorescence, such as can be seen in the inferior side. But other regions, it appears that the RPE is quite well melanized and doing okay, but the outer segments are not. So that's interesting to start with. And that is maybe not terribly unexpected because these are the regions where the OCT is kind of not as dramatically abnormal as some of these regions down below. Here's another example, switches the retention vertically upside down in this case, where the greater disease is in the superior area and less disease is in the inferior area. And again, the outer segment maps show some correspondence with the autofluorescence in some directions and less correspondence in other directions where there is a homogeneous retention of RPE, but loss of outer segments. One of our key interests is progression, and here's an example of performing the same analysis in the same patient over 2 years. And we have quite a bit of data over 2 years collected in these patients. And here's a pair of autofluorescence images 2 years apart, and one can see that there is a clearly progression coming in from peripherally inwards at these in these patients, the boundaries. And similar boundaries are apparent on the OS maps over 2 years, and there is a quite dramatic progression, considering 2 years is not a very long time. When we compare these quantitatively, we are very interested in the red regions, which show some form of progression, either from questionably absent to definitely absent, or from definitely present and questionably absent into worse categories. So red means progression, and maybe not unexpectedly, all of the progression happens in the boundaries, which is why we are very interested in it. Also importantly are the purple regions, which show no progression. So they seem to be intermingled sometimes within the progression. And there is more examples of that. Here's another example of a patient showing smaller progression over time. And very importantly, right at the boundaries in the superior area, there is clearly a progression, but in these inferior and inferior nasal directions, there is no progression. So there is, at least in our minds, there is no obvious way to predict the progressing directions from the non-progressing directions. Here's yet another example, having this peripheral retention separated from the macula with a mid-peripheral or perimacular degeneration. And we can see that the central macular area shows no progression, and part of the periphery shows very large progression, whereas another part of the same peripheral region shows no progression over the same 2 years. This is the last graph I will show, which is probably key. What we are looking at is our measures of progression. And at the top is progression of any sort against distance from the fovea. And the gray line is the average progression, which is 6%. 6% of the pixels in which we have data are progressing over 2 years. And that's the average. And compared to the average, what we see is the macula is less than the average, because typically macula is retained in retinitis pigmentosa. Perimacular area is less than the average also, because that often is very degenerate and there is no progression to see. The areas of progression that are more than average are in the peripheral regions, as well as the immediately at the edges of the macula. So that's kind of somewhat understandable. The other aspect that we looked at is there a direction in terms of isotropy around the retinal clock hours in terms of progression, and this can be seen here. The gray circle is the 6% average, and what we see is in the temporal retina and the superior retina, there is greater progression than the average, as opposed to the opposite directions. And the last two plots basically say the same thing, which is, does your neighbor define your progression? And the answer is a very definite yes, and most obvious in the last plot, which is the progression in terms of number of pixels progressing in these 2 years as a function of distance from the nearest locus with the greater disease. So when you have a pixel that is very close to another pixel that is has greater disease, there is almost 50-50 chance that they will progress. That is very different than the 6% average. So in summary then, health to disease transitions across the retina are instantaneous snapshots of health to disease progression over time. The assumption of an abrupt transition likely does not reflect the underlying pathophysiology, at least in some IRDs. And the ambiguous transition zones that I concentrated on today and their immediate neighborhoods may demarcate the retinal regions having high likelihood of progression and act as efficient endpoints for clinical trials. Here's our group at the Center for Hereditary Retinal Degenerations. Most of this work was done by Alex Maruca, who's here, and he can answer all the questions. And this work was funded by the NEI. Thank you. I'll take any questions.